Hospital. It's really my pleasure to introduce both really great colleague and friend, Kamil Uludag, who's both an MR physicist and neuroscientist, very important to note that. Kamil was recruited here to Toronto to head up the Slate Family Center for Advanced MRI. His background is a PhD in physics, when he did a postdoctoral fellowship in UCSD. From there, he headed up the human brain imaging group at Max Planck Institute of Biological Cybernetics, and then moved to Maastricht, where he was head of Department of Cognitive Neuroscience. His specific field of interest or strength, I would say, is within ultra-high field MR, and also with a very strong interest in applying those things towards neuroscientific questions. And we met originally around sort of a common interest in regards to laminar processing in the human brain, which was the first time we met. And since then, it's been fantastic, as Kamil also now is involved as part of Crania, where we folded in the low field magnet into his high field portfolio to maybe round out the entire MRI program here at the Toronto Hospital. So I'm going to now let Kamil take it away and talk about laminar processing studying using fMRI.
Okay, thanks Francis and Taufik for the invitation, and thanks Taufik for the introduction. I will give an overview about fMRI modeling, or how do we understand fMRI signal in general. And I would like to get actually a lot of feedback from you, because a lot of you come from a much more microscopic level, and I would like to sort of see how we can integrate our efforts into one common framework. So that's a very simple overview about my talk. So because of the diverse background of the people, I will just give a primer what actually is functional MRI. And then I will describe our modeling and the fMRI response on a macroscopic level. So on the level of big voxels or brain areas. Then I will also, again, of diverse background, I will talk a little bit what are actually lamina in the cortex. And I will talk about our efforts in laminar fMRI, modeling laminar fMRI. We have published many papers on experimental laminar fMRI papers, but I will talk today about modeling. And this is ongoing work. So I think it would be good to hear your opinion, what you think, how we can extend that framework. So what's fMRI? So the base of fMRI was sort of already discovered in the 1930s, and then again in 1982, they discovered that actually blood hemoglobin, the oxygenation status of the hemoglobin is changing the magnetic fields. So if you have oxygenated hemoglobin and the circle is a blood vessel, then the magnetic field lines are like this. If you have deoxygenated, then the magnetic field lines change. And the reason is deoxygenated hemoglobin is paramagnetic. So which means the change in blood oxygenation changes the magnetic properties of the blood, which is not very surprising given that the color of blood changes depending on oxygenation, and color is nothing else in physics terms than electromagnetic properties. And magnetism is one of the electromagnetic properties. So the change in blood oxygenation changes in the magnetic properties of blood. That's the take-home message of this slide. So this is illustrated here. Here you see a blood vessel. In red, you see oxygenated hemoglobin. In purple, you see deoxygenated hemoglobin. And because deoxygenated hemoglobin is paramagnetic, it creates magnetic field disturbances, which is shown here, intra and extravascular. This is during baseline. During activation, so when we increase neuroactivity, then blood flow increases and washes out deoxygenated hemoglobin, which means the magnetic field disturbances are becoming less, and we are measuring an increase in the fMRI signal. The physics of it is quite complicated, which is described in my 2009 paper, but the simple message is the blood oxygenation change. It depends on blood flow, blood volume, and oxygen metabolism. So already at this image, it becomes clear with fMRI, we are not measuring neuroactivity. What we are measuring are the consequences of neuroactivity on the blood vessel level. And this was discovered in 1990, where Ogawa did a simple experiment. This is a rat brain, a cross-section. This is sort of the cortex, and then the thalamus, and obviously in rats, the cortex is much smaller than in humans. If he changed the oxygen content of the inspired air, if it was pure oxygen, 100% oxygen, you see an image like this. If now the rat was inspiring normal air, what you see, you suddenly see appearance of these dark lines. And these dark lines are veins. And the veins contain deoxygenated hemoglobin. And this was showing that the MRI signal depends on blood oxygenation. And he suggested in the 1990 paper, perhaps this effect can be used to detect neuroactivity in the human brain. And he called the signal the BOLD signal, and this stands for blood oxygenation level dependent. So this was the starting point of fMRI. So fMRI is a relatively young method. It's sort of 30 years old, approximately. And there are now many, many thousand papers per year published. So it's a big, big industry. Now, fMRI has very many properties which are very strange at first sight. One of the properties I'm showing here, this is from a paper from the Logothetis lab. In red with dots, that's local field potential. And you see, just look at the red ones. During stimulation, here in x-axis time, and in gray is the stimulation period. You see an increase in neuroactivity. There's an overshoot and goes to steady state, and then you see a post-stimulus inhibition. The fMRI signal is this other red curve. What you notice, it's from the same sort of area, but you notice the fMRI signal is much, much slower. So one question is, why is the fMRI signal so slow compared to neuroactivity, multi-unit activity, or on local field potential? And when I went to Tübingen, I suggested Nikos Logothetis to do this kind of experiments. This is a monkey brain. It's a visual cortex. Here you see the electrode. The electrode looks much bigger than it is because of stability artifacts. And that's actually one interest of Taufik, because we want to image at 0.5 Tesla and insert image electrodes in patients. And this susceptibility artifact is a problem. And also heating might be a problem. But in monkeys, so this is the electrode which is inserted. And what I suggest to Nikos was, let's measure neuroactivity and fMRI response to different level of CO2. By the way, can you see all my pointer? I hope so.
Yep.
Okay.
Yeah, we see
everything. Yeah, perfect. Yeah.
Okay, great. So for different levels of CO2, you see multi-unit activity now in yellow is the stimulation. You see the multi-unit activity goes up, steady state, and then post-stimulus inhibition. Local field potential looks very similar to this. And this for different level of CO2. CO2, as some of you might know, David Mikulicz, Joe Fisher, Jim Duffin, they're using it in Toronto Western to measure vascular reactivity. And the assumption is it doesn't change neuroactivity, but it just changes the dilation of blood vessels. And this data confirms it. The neuroactivity, at least to a certain level of CO2, remains unaffected. Then my interest was, let's look at the fMRI response. Here on this one, you see blood flow. The red one is for 0. CO2, so blood flow increases for stimulation and goes back to baseline. So nothing spectacular. However, if you go to 6% CO2, which is corresponds to 10% CO2 in humans, what you see is the baseline blood flow increases. That's this vascular reactivity effect. But there is no response to the stimulation anymore because the blood vessels are paralyzed. So that's an interesting effect. And this might be an interesting effect also for epilepsy, because we might have something similar going on in epilepsy, because when people measure fMRI in epilepsy, they often measure negative BOLD, even though neuroactivity increases. And the reason why neuroactivity, why we see negative BOLD, this is shown here in this middle panel. This is statistical maps for 0% CO2, 3% CO2, and 6% CO2. And blue means decrease in signal. And the reason why we're measuring negative blood oxygenation is, I told you that the blood oxygenation reflects blood flow, blood volume, and oxygen metabolism. And at 6% CO2, we don't have any change in flow and volume, which means the bold signal reflects just oxygen metabolism for 6% CO2. Now, the interesting effect, so this was blood flow on the left, on the right is bold signal. This is from a different experiment, therefore the simulation is a little bit shorter. Here you see the in red, the bold signal to 0% CO2, during 0% CO2, so it's very normal. With 6%, you see negative bold. Now, the punchline of this experiment is, the red line is a combination of blood flow, volume, and oxygen metabolism. The blue line is just reflecting oxygen metabolism. And I told you it was a mystery why fMRI is so slow. And when you look now, the blue line is as slow as the positive response, which means oxygen metabolism is a slow process. Or in other words, the reason why fMRI, functional MRI, is such a slow response is it's not coupled to local field potential or multi-unit activity, it's coupled to oxygen metabolism, because the blood vessels, their function is to deliver oxygen, glucose, to carry away toxic elements and carry byproducts of metabolism. And because metabolism is so slow following neuroactivity, fMRI is so slow. Or in other words, fMRI actually is a fast response, only it's coupled to oxygen metabolism and not to neuroactivity directly. To summarize the fMRI part, the neuroactivity changes blood flow, volume, and oxygenation. And combined with the effect that deoxygenated hemoglobin is paramagnetic, we are measuring MRI signal changes following neuroactivation. And in my mind, and this other people might disagree, functional MRI is primarily a metabolic marker of neuroactivity and not a marker of information processing. So that's important to keep in mind. I'm happy to answer questions during my talk, but I cannot see the chat. So if anybody has a question, just interrupt me, please. There is no question at this point. So I come to the modeling part. So usually when you look at an fMRI paper, you see images like this. You see a brain very nicely reconstructed in 3D, and you see color-coded the fMRI signal changes correlated with stimulation. So the more yellowish, the more the signal changed simultaneously with the stimulation. Another way of analyzing this data is looking at the different brain areas, like from this study with different brain areas, and look at graph models, look at models of how brain areas are interacting with each other, and using the fMRI time courses to determine which brain area influences another brain area and what the connectivity strength during that specific task is. Now, this one and this one, they assume that actually fMRI signal reflects directly neuroprocessing. And as I argued before, that's not the case. But you will find a lot of papers where they use fMRI and they will say neuronal correlates of consciousness and this kind of things, even though it's not measuring neuroactivity. Now, if you want to therefore mathematically describe the fMRI signal, we have to reconstruct the chain of events from neuroactivity to the fMRI signal. So here you see sort of a sketch of a neuron, synapse, and postsynaptic neuron astrocytes, and we have arriving spikes, and then they release neurotransmitters, and they have then postsynaptic activity. And these numbers indicate how much energy is consumed. The most energy which is consumed is postsynaptic activity, or the pumping the ions back to their state before the release of the neurotransmitters. Action potentials themselves do not consume much energy, and that's also one of the reasons why fMRI reflects local field potential, because the most energy consuming processes are the postsynaptic activity, and local field potential reflects mostly postsynaptic activity. So we call this then neuroresponse. It's a very simplified notion. This neuroresponse changes blood flow. This process is called neurovascular coupling. Then we have the hemodynamics, blood flow changes blood volume and deoxygenated hemoglobin. And then we have measuring then the deoxyhemoglobin as primarily as a bold response. And then we get the fMRI activation located in the brain. So if you want to model, if you want to therefore make inferences from the bold signal, from the fMRI signal to the level of neuroactivity, what we have to describe are the different transformations. So one would be the how do we describe neuroactivity? How do we describe neurovascular coupling? How do we describe the hemodynamics? And then how is then this change in the deoxyhemoglobin translate in the fMRI signal? I will not talk about this because it's a big topic. This would be a separate lecture. So that's MRI physics. So I will talk about physiology 1, 2, 3. And in developing this model, we were guided by experimental results. And the experimental results we were guided is, I show you just examples. This is again from the Logitidis paper. This is in gray, you see neuroactivity, overshoot, and posthumous inhibition. In blue and red is the bold response, much slower as seen. And the idea is the bold response coupled to local field potential, and local field potential is a result of excitatory and inhibitory activity. And the new response can be not always like this. They can be also negative. They can also have different time courses. So in blue, for example, you have an initial overshoot only in the negative direction, and then it goes down to steady state, but much faster and much lower level. So new response can have very different forms. And from the hemodynamics, in red, you see the bold signal. In black, you see the blood volume. In blue, you see blood flow. And you see here blood flow and blood volume are different, which means the model has to describe that blood flow and blood volume can have different time courses. So now these three experiments I will now formalize in mathematical equations, but I will not go too deep into the mathematical equations themselves, but explain sort of the ideas behind it. So this is now the new response. In the new response, we have E, the excitatory activity, and I, inhibitory activity, and they are coupled and with a certain coupling strength. And at the same time, we have an adaptation effect described by lambda. So that's our neuronal model. And the E is the excitatory activity primarily reflects or predominantly reflects is reflected in local field potential. And the excitatory activity from one brain area interacts with excitatory activity in a different brain area. And this is described by these equations. And this is described in more details in our papers in 2015, 2017. The neurovascular coupling, we assume neurovascular coupling is strictly feed forward, which means neurons are active, they release chemicals, and the chemicals act on blood vessels, on the muscles of blood vessels. And the blood vessels then respond, if neuroactivity increases, responds by dilating and delivering more blood to the tissue. And we assume that the neurovascular coupling is strictly feed forward. And I think this is true for healthy physiology within the standard physiological range. Some people assume that there's a feedback element, which means how much the blood vessels dilate depends on how much they are already dilated, which means for the same neuroactivity change, you can get different blood flow change depending on the baseline level. I don't think this is true, but this is a controversy in the literature. And one point to keep in mind, this might be violating diseases. For example, epilepsy, I think this the neurovascular coupling is affected. Then I argue that blood flow and blood volume can be uncoupled, which means if the outflow is delayed, then you have a blood flow and blood volume uncoupled, and this is called the balloon effect. Now, putting all these things together, we can now create sort of time courses. We can assume neuroactivity for short stimulus duration can have different time courses, blood flow response, which is like a smooth version of neuroactivity, and then the bold response, which is measured in fMRI signal. I'm not going into the details of the features, as they are very rich, and it's very interesting to look at these, but I think this will go too far away, but I'm happy to answer questions. For more block design, the new response would have these different possibilities. As you can already see, this looks like the local field potential measured in the papers of Logothetis.
I might have a question, actually.
Yes.
That's overly simplifying everything, of course, but is it a good rationale to think of MRI as being, fMRI, sorry, as being a, you know, the neural response convoluted with an exponential, like an exponential kernel? You think this analogy works in this context?
Yes, I think I'm perhaps an optimist, but I think that the hemodynamic response is actually faithfully reflecting neuroactivity, whatever this is, but in a smooth version of it, which means there's not if you have, for example, if you think neurovascular coupling is not feed forward, then you will get oscillatory behavior in blood flow, which is not present in neuroactivity. So I'm an optimist, and I think the data so far, what I have seen, and we have measured many data using blood flow, is sort of supports the notion that if you take neuroactivity, local field potentials, and smooth it, then you are getting blood flow. In the Bolt signal, because you have the blood volume effects, the Bolt signal has additional transients which are not present in the neuroactivity and blood flow. So therefore, the blood flow is, I think, quite a good reflection. The Bolt signal is more complex.
Okay, cool, thanks.
Now, this was not the the generative model, how we are modeling in a feed-forward way from neuroactivity to the Bolt signal. We can also use it to analyze data by using model inversion. I'm just showing one example. This is an experiment what we did. You have a hemifield stimulation checkerboard, and at the same time motor activity left and right. This activates 5 brain areas, or more than 5 areas, but this, for illustration, just I'm assuming 5. V1 left and right, driven by the visual activity, visual stimulation, and 1 SMA by the motor responses. Now, then if we have the data and if we extract the time courses, we can insert the time courses into this formalism and invert the signal using Bayesian Model Inversion and get to the excitatory and inhibitory neuroactivity. And if we do this, then we can determine how the different brain areas are connected. What are the connectivity strengths? What you see is published in 2017, and there are many more details about this process. And you see, based on this data, V1 is activated first, and then it sends signal to SMA. There's also some SMA to M1 activation, but it was below threshold. And the arrows are showing the connectivity strength. Now, the important thing is, because fMRI is an indirect reflection of neuroactivity, this result is very much model dependent. So if you would assume, for example, a different model here in the neurovascular coupling or blood flow, blood volume, then we are getting actually a different connectivity profile. And this is shown here. This is if you use the Carl Friston's dynamic causal model, use the same data, what you are getting is a connectivity profile between brain areas which look very different. For example, we are getting inhibition from SMA to V1. This is shown by the blue arrows going from SMA to V1. And the reason why this is the case is their hemodynamic model does not explain time course very well, and it tries to therefore fit the unexplained variance by inducing or by assuming connectivity between brain areas which are not there. And actually, we have Carl Friston is called on these papers, on our papers, and he agrees with our statement that all physiological assumptions is more appropriate to fMRI data than their original paper from 2003. But this is important to keep in mind because fMRI is an indirect reflection of neuroactivity. When we calculate neuroactivity from fMRI, we have a model dependency. So for different models will give different results. That's sort of a bad news, but if this is the reality, then we have to deal with that. To summarize the model, so in the on the neural level, we have excitation inhibition, and the excitation between two brain areas interact with each other. Then we have the excitation releases vasoactive signal, which changes blood flow in a feed-forward manner. And then we have hemodynamics, which then translate into the Bolt signal. Of course, as Jeremy has said, this is simplification because we have also direct contribution of inhibition to neurovascular coupling, but we have to balance model complexity because we don't want to just generate a model which can predict the Bolt signal, but we want to use Bolt signal in inverted. So if we introduce more parameters, then the model invertibility will suffer. And I think it's a good approximation. The reason why I think it's a good approximation, there are many reasons, but one of the reasons is because we have developed the model in a way that the excitatory activity reflects local field potentials, and local field potential is most correlated with the Bolt signal. I think this approach is good. However, I would very easily admit that this neural part can be improved, and we are actually working on improving this. So I will go now to the third topic. If there are any questions, please interrupt me to the first and second topic.
I have a quick question, Kamil.
Yes.
If you're taking them. So this is John. So this came up for me just after you described the physiological model, which you have the inhibitory neural population in, which is deviating from some of the at least kind of classic original conventional perhaps ways of modeling neural activity at the time related to fMRI. But the question is like, what is the contribution of the inhibitory population to the dynamics on the time scale that you're seeing in the Bolt, like on the time scale of tens of seconds?
Yes, so the inhibition has very, very strong influence on the Bolt signal. However, it's sort of indirect via the excitation. So that's there is obviously a metabolic cost of inhibition, and this is approximate to be 10 to 20%. So we could actually very easily change it. If you go back to this model and then make a link from inhibition to the blood flow and just have a weighting of 20%. The reason why we don't want to introduce an additional parameter is the model becomes less invertible, which means you have then correlation between the parameters you're inverting. And as long as we don't have any strong data we can base on this, I would not like to do that. But I sort of for the future, I think this sort of the vascular part and the MRI physics part, I don't see many big changes coming into the future because we are dealing with macroscopic signal and we have enough data to be sure that this part is correct. But the neural activity, I think, can be made much more complex, keeping in mind the model has to be invertible. So I think inhibition has very strong influence. In the current model, it's an indirect influence, but it has strong influence.
Okay, kind of phenomenologically, does it kind of make the curves kind of saturate slower or something?
Yeah,
like that?
For example, if you would not have inhibition, because in the DCM version of 2003 they don't have inhibition. Yeah. There, I have not shown this, but it's shown in our 2015 paper. The phenomenology of neural response is very, very limited. If you don't have inhibition in it, you don't generate a wide family of neural responses. So inhibition, therefore, is essential in order to create neural responses which are more realistic. So in that sense, inhibition is very for us. That's one of our, I would say, key inventions compared to the 2003 Carl Friston paper. But in terms of metabolism, it's sort of an indirect influence. So, but as in terms of phenomenology, without inhibition, you cannot explain the Bolt signal.
That's great. Thanks.
Okay. Thanks for your question.
Could I ask just a quick clarification?
Yes.
Yeah, so in your previous slide, when you showed the dynamic causal modeling, this one. when you made the statement about it being model dependent, sure. But in the sense, if I understood, like there's the model, the whole cycle model that you mentioned, and then there's sort of the dynamic causal model where one is sort of imposing what one thinks is happening, right? So, I mean, it's dependent on, the different structures and what connectivity that one is just trying to fit. So is that what you meant when you said model dependent, as opposed to?
No, so in both cases, we assume the same graphical model. So we assume these 5 areas interact, and now we are using the physiological model in order to determine the connectivity strength between these brain areas. So that the graphical model or the hypothesis we want to test is the same.
Okay.
But the connectivity strength, what we are getting out, is depending on what physiological model we have used.
Okay, got it.
Now, this was sort of bad news that it's model dependent. There's also good news. If you have several conditions which are sort of similar, then all of the effects which are, you see here, the differences, they actually cancel out, because the unexplained variance in this one is also unexplained variance in the other condition. And therefore, the connectivity, which is sort of artificially introduced between brain areas based on the wrong model, they sort of cancel out. So therefore, one of the recommendations, where what also Carl Friss and Klaas and Stefan agree, DCM is much more, dynamic causal modeling is much more robust if you use stimulus condition differences instead of single conditions. The main difference, therefore, is our model, because it explains much better the time courses, is also much more robust for single conditions. And if you have condition difference which are more apart than from each other, then also you cannot assume that the artificial estimate connectivity is canceling out. So therefore, in that sense, there is good news that condition difference are more robust. But still, if you have the better model, then you should use it. And we have done model comparison between these 2 approaches, and that's all described in the papers.
Perfect. Thank you very much.
Okay, I will be a little bit faster now because I'm running out of time. So introduction, what are lamina in the cortex? One is, so this model is very macroscopic. It deals with the brain areas on the level of large voxels. If you, however, go to higher resolution, what you see is in the cortex, you see lamination, which means this, for example, is cell density and myelination. You see differences in the cortex. And if you zoom in into one, this now cell densities, you see different layers. And if you blur your eyes, you can actually identify layers relatively easily. For example, here you see less cells, and then here you see high cell density, and there are mathematical methods to identify layers from this kind of images. And that's the people who have done this most are Carl Zillis and Katrin Amundsen. And if you look at different brain areas, you see the profiles, layer profiles in cell density differs in which brain areas you are. Now, based on cell densities and based on how synapses are distributed and connected, Douglas and Martin and other people have developed microcircuit models. I will not go too deep into the microcircuit models, and they are experts in the audience. The sort of very simplified view is it's obviously very much simplification compared to reality, but the idea is layer 4 receives feed-forward input from thalamus, then the signal is distributed, then you have long-range cortical targets and feedback, and their synapses are distributed differently than the input to the layer. In fMRI, we make a very, because our resolution is not that good, that we can identify all these different processes, neither spatially nor temporally. We make often the assumption when we see activation layer 4, that reflects input. When we see activation in layer 3, 2, and layer 5, 6, this reflects interaction with other brain areas and feedback. And I would like to hear your opinion how wrong this assumption is, what layer fMRI people are doing. But this is currently sort of the way lay fMRI people think. Obviously, this based on this, we know that the microcircuits are also depending on brain area. Here's, for example, microcircuit shown for hippocampus. For prefrontal cortex, again, we have a different microcircuit, and that's from a paper from Emily Finn. So the neurocircuitry is depending on brain area investigated. Now, when people do laminar investigations, what they usually do is they insert an electrode and with having different contact points, and they extract laminar neuroactivity. Obviously, we cannot do this in healthy human subjects. You cannot stick an electrode. So usually laminar, the field of laminar function activity is dominated by invasive animal electrophysiology. However, in patients, now there are some groups who are measuring laminar electrophysiologic profiles in human patients. I just want to show just one example. I don't want to go too deep into this example. This is from Alexander Meyer's lab, where they measured now CSF is on top, white matter is on the bottom, and x-axis is time. And this is for two different simulations. What you see in this cortical current source density, you see two different profiles, laminar profiles. Here you see a peak in the top and a negative peak in the bottom, whereas in the other condition, you see a positive peak where there was no peak at all. So to just the take-home message from this slide is laminar profiles on the electrophysiological change depending on stimulus condition. And the hope is if you know the laminar profiles, we can actually infer something about the processing streams of in that patch of cortex. When we would just look into electrophysiological response average over all layers, we would not be able, we would say how much it's active, but we would not be able to differentiate different processing streams like feedback versus feed-forward. That's at least the hope. To summarize the lamina, we have cortical thickness, which is usually 2 to 4 millimeters thick. We have cellular distribution, connectivity, and receptor architecture, which are brain area specific. There's a very influential canonical microcircuit, but which describes processing streams in cortical areas, but it is mostly developed in cat models and also macaque models, but it's not actually not for all brain areas, not for all species. And there are strong indications that the microcircuit is actually also brain area dependent. And laminar neuroactivity is typically measured with electrophysiology. Now I come to laminar fMRI. Laminar fMRI is a relatively new field. This is a number of papers published per year. As you can see, till 2015, we had like 2-3 papers per year. Now, 2020, we had 43 papers per year, which means probably this year will be one publication per week, which is quite a lot compared to previous year, but obviously it's still a small field because compared to, I think, 3,000-4,000 papers using fMRI per year, compared to that, it's still a very small field. But it's a very dynamic field and very advanced group are working on this, and it continues to grow, especially with the delivery of 7 Tesla MRI scanners. Now, I described you the microcircuit model and the fMRI approaches. You have sort of a nuclei, like LGN projected to the middle layers mostly, and then it goes to superficial layers, say in V1, and then it projects to the next brain area, again to the middle layer, and then if there's feedback, then it goes to superficial and deep layers. So again, this is a very simplified view, and I would be very happy to collaborate with you to develop more sophisticated approaches compatible with laminar fMRI. Now, I told you that the cortical thickness around 2 to 4 millimeters thick. In standard fMRI, the voxels are approximately 3 millimeters thick. So which means a voxel is now outlined here. We can actually not look into different layers in the brain using standard fMRI approaches. So we cannot look output, input, and feedback in the brain with standard fMRI voxels. because the cortical thickness is approximately in the same dimension as the voxel dimensions in fMRI. If we go to higher resolution, let's say we have now 5 times higher resolution, then we actually can hope that we can distinguish, say, top layers from middle layers and bottom layers. I want to point out, if we go, say, to 5 times higher, in 3 dimensions, this 125 times higher. So laminar fMRI is pushing sort of the limits. We have 50 to, typically we have 50 to 100 times higher spatial resolution compared to standard fMRI. So that's quite something. And when I started laminar fMRI, I did not know, and a lot of people were asking me, is laminar fMRI at all possible if you go to 100 times higher resolution? And the answer is, this is from Shiranga Kashyap, my former PhD student, and soon postdoc again in my lab. What you see here is sort of coronal slices here, and you see in color-coded statistical activation. And to my surprise and surprise of a lot of people, we are detecting very robust activation in single runs going to that high spatial resolution. Compared to electrophysiology and microscopic methods, this is still bad resolution, but compared to what is standard in fMRI, this is very high resolution. Now, I was talking with Francis before the talk. Why would somebody want to do fMRI at all if 0.8 or 0.7 millimeter is considered high spatial resolution and pushing the limits? Even though electrophysiology, we can go to much higher resolution. And the answer is, it's non-invasive, which means we can do it on health subjects. We can put the subject in and you get a result in 10 minutes. So that's one big advantage. The second advantage is we can cover much larger patch of cortex. With electrophysiology, at least with standard electrophysiology, you have one electrode in one location. Now we can look at a large part of the brain, and we can look at brain interaction. So, which means fMRI has limitations, spatially and temporally, and also that is a metabolic marker. However, it has advantages that's non-invasive, and we are covering a large part of the brain, and we can do experiments. Whatever brain area we want to study, we can study. If we want to study prefrontal cortex, we can study. We don't have to drill a hole there. If we want to study hippocampus, we can study hippocampus or amygdala. So these are the advantages of fMRI. So therefore, I think there is a place for fMRI, but at the same time, we have to recognize its limitations. So on the neural level, we have this complex architecture of pyramidal cells, inhibitory cells, feedback, feed-forward, and so on. And we have the microcircuit model. And if you go to the vascular level, we have then the arteries and veins and so on. And this has been just accepted in Progress in Neurobiology, where this model is described in more. And the main domain or the main method to access is electrophysiology, whereas this laminar fMRI. So therefore, if you want to connect laminar fMRI to electrophysiology, we have to connect the left picture with the right picture. Now, I already have done this for the microscopic level. For the mesoscopic level, we have to be more sophisticated. One experimental observation, if we look at fMRI signal on the vascular level, is like this. Now, fMRI people display data in a different way. White matter is on the left, and CSF is on the right, whereas in electrophysiology, CSF is top and white matter is bottom. And what you see is the fMRI signal as a function of cortical depth. And what is a common observance is the fMRI signal increase from white matter to CSF. If you look at the electrophysiology, again, CSF is top, white matter is bottom. It doesn't look like the fMRI signal. And which means it's fMRI signal at first glance is incongruent with electrophysiological profiles. But there's a good reason for it. If you look at this model again, there we had for one brain area, one model. However, if you go to cortical thickness, so this is surface of the brain and this is white matter, and we are measuring deoxygenated hemoglobin. What we are measuring is at the end what's going on in these veins. So imagine you have activation in the middle layer, neuroactivation. Then you will measure fMRI activation in the middle layer, but also because deoxygenated hemoglobin is transported to the top to be drained, you will also measure deoxygenated hemoglobin changes in the upper layers, or fMRI signal is blurred towards the surface. This can be described by mass balance equation. This is in the 2019 paper. It looks complicated, but at the end, it's just mass balance, which means if blood flow goes in from left to right, then the vein is transporting the oxyhemoglobin from bottom to top, which means for each layer, you have two inputs in terms of blood flow and the oxyhemoglobin from the local population and from the population below that layer. If you use this model, we can do many, many predictions. One of the predictions is if you look at now the bolt signal as a function of cortical depth, you have here in blue activation in one layer, let's say layer 6. You measure fMRI activity in layer 6, but you also measure activity in layer 5, layer 4, and so on. Now, which means this incongruency between fMRI and electrophysiology is actually expected. And we understand why they have to be incongruent. So it would be actually bad if they would be congruent, because this would mean we don't understand what we are measuring. However, if you understand it, then we can use that knowledge to go back to neuroactivity. And the way we would do is, this is from a paper we have published, from white matter to CSF. And these are different conditions. Again, the signal increased from white matter to CSF. We can now sequentially subtract the activity from one layer to the next and get to a profile which looks like this, which should have removed the ascending vein effect. And what you see is actually, we see a peak in the middle layers, which looks for feed-forward stimulation, much more like what we would expect from electrophysiology. So I think even though fMRI, laminar fMRI does not look like electrophysiology, with clever analysis techniques, we can actually hope to get back to the electrophysiological profiles. To summarize, the macroscopic model was excitation inhibition, neurovascular coupling, and then a hemodynamic model. And then we are measuring the bolt response. The mesoscopic model, we have excitation inhibition in each layer. And each layer is connected to a similar model like this, neurovascular coupling. And then we have connected all the different layers via the ascending veins in the cortex. Now we have an ongoing NIH grant where we want to we are measuring laminar fMRI and electrophysiology in patients, not simultaneously, but using the same stimulation paradigm, which means we are measuring laminar electrophysiology in patients, and the same patients with the same experimental design go to the MRI scanner, and we are measuring laminar fMRI. And with that, we want to see whether we can link fMRI signal to electrophysiology using the model. Of course, again, I said I expect that the neuronal model is the one where I think the most development will happen. This also will be the case on the mesoscopic level. What we are working on now, we are trying to implement a microcircuit model. So substitute excitation inhibition of each layer with a microcircuit model where we have excitatory cells here where the input arise. We have pyramidal cells, 2 and 4, and we have inhibitory cells which connect the different layers. So this is initial, and this is my PhD student, Sayan, working on this. This is in its initial stages, and this kind of model is actually used, developed by the Carl Friston group, and John Griffith actually is an expert in this. He and others are working on dynamic causal modeling using for EEG, and we are trying to link laminar fMRI with dynamic causal modeling using EEG, but also use other microcircuit models and try to see whether we can make fMRI, laminar fMRI, and electrophysiology in agreement with each other. With that, I'm at the end of my talk. Compared to standard models of fMRI, high-resolution fMRI needs to consider the transport of deoxygenated hemoglobin via ascending veins. And we have developed a vascular model based on mass balance principles. And so far, using this model, it looks like it works, but the database is quite thin. And we are trying to implement microcircuit model for fMRI. And we need more data with electrophysiology and high resolution fMRI at the same time. If you would use EEG or laminar electrophysiology in patients or in animal models in the same subjects, but using the same paradigm. And I'm finished with my talk. If I have 2-3 more minutes more time, I would then talk a little bit about what my plans are at UHN. Francis, Taufik, do I have 2-3 more minutes?
Absolutely, go ahead. Thanks.
Okay,
Camille.
I have today presented just the modeling part, but my research group deals with much more. And because I'm new at UHN and a lot of the people in the audience are at UHN or CAMH and other institutions, I thought it might be perhaps a good idea to present what I will be working on. One of my main topics is MRI physics, because at the end I am an MRI physicist. We are developing new approaches for the 3 Tesla MRI. One of our focus is fast imaging methods. So we want to acquire the same images, say an anatomic image, which takes 8 minutes. We want to acquire it in 2 to 3 minutes without sacrificing image quality. Second is we want to make MRI more robust. For example, if subjects move, then the image look bad. So we want to be able to correct for motion, but not in a time series way, but in one single image. So if an image takes 8 minutes and there was motion in between, we want to create image how it would look like without that motion. Also characterizing magnetic fields and distortion. Obviously, distortion is very important for neurosurgery, because if you cannot see the target of neurosurgery well in the right location, then this is bad. So we want to also be able to correct for distortions. I'm working much on analysis, so dynamic connectivity and single subject fMRI. And also laminar fMRI, obviously, I will continue. And I'm working on clinical applications of perfusion MRI, theory and experiments. And Angelica and Jacob in my group are working on this. So measuring perfusion MRI and extracting more information about the state of the brain tissue in tumors, in vascular disease, and so on. I'm also, as everybody else, works on AI. And one of the things where actually I collaborate with Taufik is how can we translate images from 0.5 Tesla to 1.5 or 3 Tesla and 7 Tesla and vice versa. So if we acquire images at 1.5 Tesla to generate artificial images, how they would look like at 3 Tesla. And characterize fMRI brain activity using deep learning. So because fMRI is very, very complex because we have so many voxels, and we are looking into whether we can use deep learning to characterize brain activity in a better way. And they are Sean and Labib in my group are working on this. And I'm a hobby cognitive neuroscientist, so I'm continuing to do neuroscience. We are using fMRI using naturalistic stimuli like movies and video games that links to the dynamic connectivity and single subject fMRI. And we are acquiring big data, measuring human cognitive process widely as possible at 7 Tesla, not in Toronto because we don't have a 7 Tesla yet, but I'm doing this in Korea. And with that, I thank you for your attention. Most of the work I've shown was from Martin Havlicek and Sriranga. And this is my group in Toronto, which is continuing to grow. And you see all are smiling, so they're all happy. And I thank you for your attention.
That's awesome. Thanks, Camille. Great overview, great talk, very clear. Does anybody have more questions for Camille?
Hi, Camille, this is David. That was terrific presentation. Just wanted to know, with the investigation in the cortical layers in healthy people, will we be able to apply this in disease states where the vessels may be abnormal?
You mean laminar fMRI?
Yeah.
Yeah, it means usually in laminar fMRI, because we are pushing the resolution so much, our subjects are well-trained subjects. So in patients, what are the patients, the movement will be the main problem. If patients do not move, I think we can go to laminar MRI anatomically and also laminar fMRI, but the movement will be the main obstacle to translate these approaches into patients.
Yeah, I was just wondering though, if in certain disease states, so patients with dementia, vascular dementia, they may have abnormal vessels to begin with. So their bulb responses may vary because of that. So will that be a limitation?
It will be a limitation, but the approaches we are using with vascular reactivity, we can actually characterize probably the vascular architecture in that high resolution. So if there are differences in vascular reactivity and vascular architecture, we would hope that using resting state or using hypercapnia or hypoxia, we would be able to determine that.
Okay, great.
So,
So Kamil, I have a question for you.
yes.
So, in epilepsy, the goal is localization in regards to the epileptogenic focus. And a little while back, actually, David was part of this. We had looked at cerebral vascular reactivity in epilepsy patients, particularly temporal lobe patients. We didn't see a lot, admittedly, in regards to the differences between the two sides. We selected patients who had just one epileptogenic zone in the temporal lobe. We know that there is things like interictal hypometabolism in patients when you look at their PET studies. Of course, ictal hypermetabolism. Do you think these kind of methods, particularly the modeling component of it, or more sophisticated ways of implementing CVR, given that you understand more now about where the signal is coming from, as a tool for localization of the epileptogenic zone?
I think so, and also to sort of stratify the population, because not all epileptic patients will be the same. So, and there are differences in vascular reactivity and so on, might be actually quite relevant. So I think having a good scanner where the data is good, because data has to be good, let's say have higher resolution anatomically and functionally, will be important. But I would say you need a battery of measurements. Vascular reactivity is one, because sometimes the vascular reactivity might look fine, but the response to neuronal stimulation might be different because the neurovascular coupling is disturbed. Because vascular reactivity is sort of before is sort of after neurovascular coupling. So vascular reactivity might be fine, neurovascular coupling might be disturbed. Or it can be the other way around. Neurovascular coupling might be fine, but vascular reactivity might be disturbed. So we have different options. So therefore, to have a comprehensive approach where you have anatomical measurements, where you might see lesions just visually, having high resolution and high sensitive approaches with good contrast, but also having functional stimulation where you evoke neuroactivation in that area, and vascular reactivity, CVR. I think to have such a comprehensive data set and look at individual differences and population differences to control subjects will be, I think, very interesting. And I'm quite sure you will see something from there. I don't think anybody has really systematically done this.
Yeah, that'd be something. I mean, for us, because we're kind of like a new program, the whole PET side of things never really evolved very much. And so, we're always looking for other localizing tests. So this would be fantastic.
And the good thing, that's what I also point out with fMRI, is it's really non-invasive. You can do it relatively quickly. It means if you have the right experimental designs, getting anatomy, functional, CVR, not that it's simple and very, very quick, but it's still all, if you have implemented them well, it's you can standardize this quite, and you can do this in many, many subjects.
That's great. Kamil, there's a question from Bill Hutchison. Is there a known relationship between LFP oscillatory activity and fMRI or laminar fMRI known?
So the oscillatory activity to fMRI is actually, I think, very complex and understudied. So one of the goals of the DCM for EEG is actually to predict oscillatory activity. And by using our approach with the hemodynamics, we would actually be trying to address it. Because I think if my sort of hypothesis is correct, that fMRI is a metabolic marker, then you have to ask yourself, how much metabolic costs are associated with oscillations, and what are oscillations a marker of? And I think there will not be a general answer. So there will not be an answer like this oscillatory frequency is always coupled positively with fMRI, and this oscillatory activity negatively, it will be very much context dependent. So, so from my point of view, I think it will be difficult to have one-to-one relationship, but it's an understudied area, and I hope that we will be able, with this advanced modeling, to address at least some of the questions.
Thank you. I was wondering, maybe not so much with frequency, but with power of oscillations, presumably a higher power at the same frequency. Same. Okay.
It's the same argument, because the synchronicity at this moment, let's assume you have the same amount of neuroactivity and same amount of action potential and so on, but the synchronicity changes. Now, synchronicity change will then increase power in a certain frequency, because they're not smeared out in all the frequency bands. Now, I think synchronicity changes will have a metabolic effect, because if you have two neurons at the same time firing, then the reversal potential for pumping back the ions will be different. Because let's say you have excitation inhibition at the same time, then from the electric point of view, you're transporting zero current. That's as much easier than if you transport a positive current or negative current. So the ion channels have to work less for if excitation inhibition are synchronized. So therefore, I think both power and frequency will have a complex relationship to metabolism, and it might be very context dependent. So at this moment, I think we need more data. That's modeling will help, but at the end, we don't have enough data to relate it to parameters or to domains with each other.
Thank you.
Just in the sake of time, I think Mary Pat, you had a comment you wanted to make?
Yeah, actually, thanks a lot, Kamil. Great talk. Both comment and a question. So I think you probably know that we actually have probably about 10 years of epilepsy data on the GE with both resting state and task activation. So I invite any of your friends who want to come and play with us to do so. But my one question really had to do with, because a couple of times you made the comment about deranged neurovascular coupling in epilepsy. And I've wondered about that. And I've also wondered about whether it has anything to do with epilepsy versus medications that people are on. But what's the basic foundational evidence for that? And is it just in the epileptogenic zone, or is it, throughout, et cetera? What can you point me to on that?
Yeah, so medication is a big issue. So all the things what I've described, like neurovascular coupling and that it's feed forward and so on, medication will have influence. Mm-hmm. So therefore, even coffee, if you scan the same subject with or without coffee, you will not change the activation pattern, but you will change the amplitude. Mm-hmm. So fMRI amplitude is sort of very sensitive to medication, for example, and it might be also pattern might be also very sensitive. Now, my evidence for not in all epileptic patients neurovascular coupling will be disturbed, but the reason why I think in some it is in some epileptic patients where you have strong, we have epileptic activity in the scanner, you are seeing negative bold. So you have an increase in neuroactivity, strong increase in neuroactivity. However, you have a decrease in the fMRI signal. We usually interpret decrease in fMRI signal and decrease in neuroactivity. So how can it be that we have an increase in neuroactivity but a decrease in the fMRI signal? And one of the possibilities is a disturbed neurovascular coupling. Like if you have too much CO2, then you paralyze the blood vessels and you get negative bold. In the example with the monkey, what I've shown. Another possibility might be the activity is so strong, the blood flow is sort of maxed out. Mm-hmm. You don't get any blood flow. Then again, this would then be the neurovascular coupling would be intact, but the hemodynamics would be not intact. Right. So therefore, I think I would not say in all, and also not in all brain areas, but there's enough data to show that we can only explain some of the data when we have something in that chain of events, what I've shown from neuroactivity bold, something is disturbed.
And this is from the kind of combined GE-fMRI
Yes, yeah.
studies, yeah,
Yeah.
which we'll also be able to do because we have equipment. So thanks.
Okay, I'm just taking over here because Taufik had to head off. So maybe we'll just take the last question that I see here from John Griffiths. Go ahead, John.
Thanks, Francis. Great talk, Kamil. Thanks. Yeah, quick comment and a question. The quick comment is it's great to see that you're thinking about these models, these microcircuit models, thinking about them from the point of view of fMRI and electrophysiology. And the comment is that it's sometimes crazy to me that it's taken so long for that to, it hasn't really kicked off that as a kind of real area of study, but people were talking about this in 2007, like, and before then, right? So it's kind of curious that there hasn't been more action on that, but science is hard. The question is in that respect. So if you're interested in having a robust experimental model that you can study from the perspective of electrophysiology and from high resolution fMRI, what are your go-to experimental paradigms for that? And I guess they're going to be like low-level, simple, robust visual and auditory sensory paradigms. Like, what's your kind of favorite robust paradigms for inducing consistent responses in fMRI and electrophysiology tasks?
Yeah, I think low-level sensory is good for methods development, but they are neuroscientifically not very interesting. So what you would probably need two conditions where you manipulate a higher cognitive process, which has influence in that brain area. So you can have one condition which is sort of sensory driven, but you would need another condition to, as a contrast, because I think the more variation you have, the better it is, given if that you still have enough signal to noise to analyze, because laminar fMRI has not a bad signal to noise, so therefore too much variation will counteract your goal. But I think just sensor driven would probably not be sufficient. I would have it as a tool in my sort of in my reservoir, but as a only unique, probably it will be insufficient. You would need cognitive. So you need at the end manipulate the laminar profile in a certain way, which means if you have a feed-forward stimulation where you get a certain laminar profile, you need a contrasting condition where you have a different laminar profile, either in electrophysiology or in fMRI or in both. Otherwise, if you don't have variation, everything will be correlated with everything. Does this answer your question? I'm not sure.
Yeah. I mean, I definitely agree. On the other hand, I think about like MEG, for example. Like MEG people, they really like to tap someone's hand, right?
Yeah. It means I do most of my experiments, because I'm a methods person, are very, very simple. I'm not interested in checkerboards in my real life, but I'm using checkerboards in methods development because it's very robust. But I think if I want to relate EEG or MEG to fMRI, we need more sophisticated approaches, because we need to vary. We need sort of span a space of oscillations of electrophysiology activity and fMRI activity to break down simple, trivial correlations. And that's, I think, therefore important. For just EEG or MEG methods development or fMRI methods development on their own, simple stimuli are preferred because they are robust. But if I want to relate these two, you need to span a space of possibilities.
Yeah, great. That's a really interesting perspective. Yeah, really looking forward to seeing the action on this front. And like I said, I think that the concurrent, or at least the joint electrophysiology and fMRI models, I mean, we've got to go in that direction. And
yeah,
I feel like that's kind of the frontier for the 2020s of neuroimaging, is trying to kind of stitch together fast and slow time scales.
Yeah, let's talk about experiments then.
Yeah.
Okay.
Thanks.
Okay, thank you. And Taufik had to run off. So I have the pleasure of thanking Kamil to give us this exciting talk. And I just want to mention that it is being recorded. So those people who came in late, or, you have colleagues who couldn't make it for whatever reason, in the coming days, just go to the KCN Hub YouTube and it should be available here, because I know it was very fantastic for my expansion of understanding. And look, I saw you had microcircuits in there, so maybe down the road, but it's really cool and exciting. So look forward to meeting in person in the not too distant future. So thank you.
Yeah, I hope so.
All right. Thank you.
Thanks everybody for attending. Bye-bye.
Bye-bye.